9 and 10 lectures are all on D2L already. I have sent them to the
10.
printer. I have 9. We're hoping to get 10 soon. So we'll see. But chapter 8, recombinant DNA technology. So this is really big in the biotech industry. Being able to use cells and biological molecules to solve problems or to make useful products. A definition for biotechnology is the use of any living organism or subunit of some organism to better serve us as humans, basically. So people get kind of scared going, no, biotech is new. It really isn't. If you think about it, beer is biotech because we are using a microbe to make beer, to ferment. And beer's been made for how many thousands of years? So, just getting bread to rise. And so biotechnology is not actually new. How we're using it now, we're having all sorts of new approaches. But this is where we get a lot of our antibodies. So our drugs come from biotechnology. Okay, so some applications. Recombinant DNA technology. So recombinant DNA. The whole idea with recombinant DNA is that you're taking DNA from one source and putting it into another source. So Genentech is really known for this for their humulin. So it used to be that people who had diabetes, they'd have to go through, they need insulin. And so you'd have to keep going through different organisms because eventually their body would become immune to that animal's insulin. So they'd have to go to a different one and a different one. Well, now Genentech's been able to clone the human gene for insulin. And so now they can produce human insulin. And so we have humulin, which patients can get that, and then they don't become immune to it because it is their own, it's a human protein. So that's one of the applications of recombinant DNA. You take a human gene and you express it. For Genentech, they do everything in CHO cells, so Chinese hamster ovary cells. But we can do pharmaceuticals, therapeutic applications, so genetic screening. You can use DNA microarrays to screen for inheritable diseases. You can identify pathogens in DNA or blood sample. DNA fingerprinting, which we'll talk about a little more later. Identifying individuals or organisms by their unique DNA sequence. Protein synthesis, so specifically cloning certain proteins or peptides that you want or need. If you can find one from like a pathogen, then you can create an antibody against it, so then it's a way to create a drug to treat somebody because you've been able to artificially create the protein in a lab. And also it can be a lot safer because you don't have to grow the pathogen to get that protein. You can grow just a little piece, just that one protein, which won't necessarily cause a disease, but it's enough to trigger an immune response. So it's how we can safely get some vaccines or antibiotic drugs. Gene therapy, they're working to treat for missing or defective genes. So the idea is there are some diseases that are caused by a single gene being not functioning properly, defective. And so it's one of those great things, like if you could just go in and replace it with a healthy gene, then all of a sudden you get rid of the disease. And so on paper, it sounds awesome. It sounds fabulous. In practical application, there's a lot of hurdles and roadblocks and things that are kind of making that harder to do. Medical diagnostics. So kind of looking at patient specimens, you can examine them for certain pathogens or gene sequences. You can just kind of see what's going on so you can help do better diagnostics. Xenotransplantation. So xenotransplantation, there's not enough human organs to donate. And so one of the things they've been looking into is animals. And so using animal cells, tissues, or organs and introducing them into body. And this is done a lot of times with pig valves for the heart. Pigs are very similar to us as far as a lot of their organs are kind of the same size, they kind of work the same way, so they're a good kind of fudge for human organs. And they've even gone in and made the pigs so that they no longer produce a protein on their cells. So we would recognize it as foreign, but now we've cloned pigs to no longer even produce that protein. So those organs now don't have as much of a rejection issue. And then vaccines, we use recombinant DNA to make those vaccines I was talking about earlier. So, and that genetic screening, I'm sure Dr. Kubo talked about the 23andMe, so being able to actually screen your SNPs is one of those little things.
Did you mention the fact that we're now able to make growth hormone and insulin in unlimited quantities?
We're now what?
Able to make growth hormone and insulin in unlimited quantities, whereas before it was
Well,
very, very
growth
difficult to come by.
hormone, so insulin was hard to come by just in general. Growth hormone was being purified from pituitary glands of cadavers.
Yeah, I know.
So it's
Very little of it.
not very much, and you also, if you're purifying it from cadavers, then you have a possibility of carrying some virus or pathogen with it. So being able to take it out of that, being able to put it into E. coli or into CHO cells or some other workhorse, you can get it cleanly, safely, and in mass quantities. Genetic recombination. So, basic idea of the genetic recombination is you are cutting a piece of DNA and you're inserting it into a plasmid or a vector. You will hear those two words interchangeably. Plasmid, vector, same basic idea. I think usually plasmids just kind of the classic sense of bacteria had plasmids. We play with vectors because we can make it do things, but either one works. So if you add a piece of your cloned DNA to a plasmid, then you now have a recombinant plasmid. And so you're able to then chemically treat, is usually the easiest way. So you can chemically treat cells, and you can make it so that your bacterial, so that the E. coli is ready to take up DNA. So you could, do calcium chloride. You do a whole heat shock thing. So you make their walls all kind of soft, the plasmid membranes open, and so then they're ready to uptake your plasmid of interest. And then, once it's in there, plasmid will replicate, multiply. It'll also produce whatever protein you put on that, your special little insert. And so every single cell following this will have your plasmid and your gene of interest. So you think of this being human insulin being put onto a plasmid, being put into a cell, and then replicated. And so now every single one of those is producing human insulin. And one of the great things about plasmids is that it's not necessarily one per cell. You can have multiple copies depending on the plasmid. Okay. So does this make sense?
Is this a picture right out of the book? I mean, it makes sense, it's just really hard to read out here.
This?
Yes.
Yeah.
On the copy I have, is this a picture out of the book?
Should be, I think so.
Okay.
I forget what figure that is. I usually look from, but yeah. Okay. So one example, common thing that is done in labs, or at least like for beginning labs, just because it's nice and pretty and it's easy, is GFP. So green fluorescent protein. You get it from jellyfish. If you've ever gone, we did in Catalina where we did a night dive. You hide your flashlight, you wave your hand, and everything fluoresces. It's the green fluorescent protein stuff. So different jellyfish, different sea creatures, different depths have different fluorescent proteins that actually fluoresce. So we can take the gene for this protein, and you can actually insert it into that plasmid, and then you end up with colonies that glow because they took up protein. So it's a very visual way of seeing that DNA was taken up by these colonies. Okay. So GFP is a great marker, but it's, it also just proves a point. Restriction enzymes. So we talked about this a little bit before. Restriction enzymes, they're always palindromes. So remember what those mean, same forward and back. EcoR1 has a recognition sequence of GAATTC. So, and then you'll either see something like this, or like this, when you're looking at it in like a textbook or in something I give you because it's just easier to draw than some of the other things. Usually it'll be this just because it's easier. And this tells you where the cleavage site is. So when you make this go full, so remember, what it matches up with, that means it would be here. And so if you just draw the cut in the same spot, draw it across, what this means is with the restriction enzyme, they come through and they cleave the phosphodiester backbone. They actually will cut this backbone. And then these are just hydrogen bonded on. So those can fall apart. There's a lot of breathing is what they call it. Anyway, so they're not always perfectly bound. So you'll end up with a G, C. Okay. This is also called a sticky end. Okay, so you can get two types of cuts. You can get a sticky end or a blunt end. And a sticky end is because these want to find partners to pair with. So it's sticky. Okay? Also, you can have a 5' or a 3' sticky end. So you go through and you label your DNA. You realize this has to be my 5', so I have a 5' overhang. Does that make sense? Why it's 5'? Or it's easier 5', 3'. So whatever's overhanging is your 5' overhang. Or if it was overhanging the other way, so you had 3' end overhanging. One of the ways I like to think about restriction enzymes is they're kind of like craft scissors. So if you're doing scrapbooking, you have all these different types of scissors. So if you cut with one pair of scissors, you can then cut something else and you can match it up perfectly. But if you cut with a different pair of scissors, those aren't going to match up right. And so each restriction enzyme is a different cut for the most part. You can find exceptions, but for the most part, each one's going to do a specific cut. So the idea is that you're breaking apart this phosphodiester backbone, getting this sticky end, and then, so if you cut your plasmid with echo R1, and you cut your piece of DNA you want to put in with echo R1, then DNA is DNA. It's not going to know that this incoming piece came from the other half of the plasmid or from your insert you're putting in there. Does that make sense? Other questions?
Did she answer what the blunt end was?
No? Okay. So, sticky ends, blunt ends. Just to make it even more clear. So we have, it's always palindromic, gets cut, gets taken apart, that's why it's a sticky end. And then you can put them together. And the thing is that, say, purple is from the plasmid, turquoise, I guess, is from whatever piece of DNA you're trying to put in. So a lot of times we PCR up fragments of DNA, cut the ends. So then now your plasmid is open with these two sticky ends. You have your piece, your insert, you want to drop in. It has the sticky ends, and you can get it so that it ligates it together. And so this is where learning how we do DNA replication helps, because we do a lot of the same tricks now, but we're doing it in test tube. So if you remember the DNA ligase, that puts together that phosphodiester backbone on what kind of fragments? What were those? Okazaki fragments. Well, that same DNA ligase can fix this phosphodiester backbone. And so you can get a closed circle again. So you can have either sticky ends or you can get a blunt end where it cuts it straight down the middle, and so there is nothing overhanging, so it's blunt. Blunt ends are great for the fact that you can mash two different blunt ends together. They're not as easy as sticky ends to put together, but there are times when that's all that works. So restriction enzymes, they're actually microorganisms, basically immune system. They didn't have a way to stop this DNA that was invading them or to break things down. So what they do is they have restriction enzymes. So the restriction enzymes recognize DNA that is not self and chew it up. And so that way it can't ever be made into into the protein that would then harm them or whatever. So this was their method of an immune system in a way. And so scientists have gone through and from all sorts of different organisms found different restriction enzymes. And so we have a huge list of them by now. We keep trying to find more. But EcoR1 is just one of the common basic ones, which is why it's always used. So, you have restriction enzymes. It's an enzyme that recognizes a specific short DNA sequence and cuts that DNA internally on both strands, okay? So it's an endonuclease, endo because it's inside, nuclease because it cuts DNA. It's very specific. It has a recognition sequence. That's what these are called. That's what the GAATTC is. That is the recognition sequence for the restriction enzyme, EcoR1. Or it's also known as a restriction site because EcoR1 will cut it. And then you use DNA ligase to join the DNA molecules together. Plasmids, okay? They're circular DNA molecules. They're found in bacteria. A lot of times they have an antibiotic resistance gene, and they can contain genes from foreign organisms. So some of the major things that a plasmid should have to be a good plasmid that we want to work with should have an origin of replication, an ORI. So this means that it can replicate on its own, autonomously. Okay, so it will be able to, once inside the host, just keep making more and more copies of the plasmid. There should be some kind of marker, usually ampicillin resistance, tetracycline, kanamycin, some kind of antibiotic marker. So this, when we're using this in a lab, allows us to transform E. coli. And E. coli, if you put it onto a plate with ampicillin, would normally die. But if it has this ampicillin-resistant marker, the gene for it, then any bacteria that has taken up your plasmid will be capable of growing on the ampicillin plate. Okay? So this is a way to select for only the bacteria that have taken up the plasmid you want. LAC-Z helps you do blue-white selection. And so this gene, when you feed it the right media, will actually produce a blue color. And a lot of times what you'll actually have a multiple cloning site inserted into that LAC-Z gene. So the idea being that that multiple cloning site is where you're going to open up the plasmid and drop your piece in. And if you've opened up that gene and dropped your DNA of interest in, you've just killed the LAC-Z site, which means you'll now have white colonies instead of blue colonies. So that gives you a way to select for bacteria that have taken up your plasmid and for the plasmids that have taken up an insert. Because you don't really want to waste your time screening plasmids that don't have your gene of interest and won't make your protein. Okay. We also want to be small. Generally we want our plasmids small, 5 kb or under kind of thing, around that size. They get too big, E. coli doesn't like to handle them and tends to shuffle them, and then that's never good for your genes. Okay. Does that make sense? Kind of the key things you'd want on a plasmid? Small, multiple cloning site, blue-white selection through LAC-Z, some kind of marker for resistance, origin of replication. So here, just another kind of going through, you can go, say we want this gene of interest. You can either go through and cleave it out of the genome and isolate it and insert it into your plasmid. Or the more common way is actually to use PCR, which we'll talk about in 8 slides or so. But same principle. Basically, somehow you want to isolate that DNA fragment. You want to cut it so that way it has specific ends, so it's going to go into your plasmid. You want to insert it insert your gene into the plasmid, and then you want to insert the plasmid with the gene into the bacteria to give us recombinant DNA. So this is how they got recombinant bovine growth hormone. So in that case, they went into the cow, because the cow already has growth hormone, took out that gene, were able to put it into E. coli, or into bacteria, usually E. coli, because it's the workhorse of biotech, and they were able to make a lot of genetically engineered bacteria that will multiply, and they'll have a ton of the BGH. So this is a way to artificially make more growth hormone. Okay? Is this making sense? Are there any
Do
questions?
they still do this now that they can make pure human growth hormone?
Yes, because they use this on cows.
Because what?
Cows.
Oh.
You want the cows to grow and produce more milk. So there's other applications. I mean, for humans, we don't use cow growth hormone, but for the cows, it's better to give them their own gene and protein. That's the whole point, is they, in the end, give it to the cow. Okay. So inserting DNA into cells. So the goal of this DNA technology is to insert DNA into cells. We've talked about before the natural methods, the transformation, the transduction, the conjugation. Right? Those all ringing bells? Okay. So artificially, there are a number of methods of doing it. Electroporation is a common, lovely method because it's nice and fast. And then you can do protoplast fusion, which is how we have gotten some of the great things like broccoli flour or whatever, the cauliflower and broccoli mixed together. You can do injections, so you can do like a gene gun. There are some bacteria, one of the cells I worked on, Cryptococcus neoformans, it has such a thick cell wall, you can't get through it. So what you actually do is you take the plasmids, coat little gold particles, and you use high pressure to shoot it through the cells. And the old method, you'd have people shooting blanks to get that pressure through. So they've come up with safer ways of doing that now. But it's a gene gun. You can actually shoot the cells and give them new genes. Or you can do some kind of micro-injections. So let me look at this. Electroporation. So you have your cell is happy. You make sure it's competent. Competent cell, that just means that it is ready to accept DNA. When it's ready to accept DNA, it is competent. So you apply an electrical field, which, so you have these cells, you've kind of chemically treated them so that they're now competent. You add a lot of your DNA to the outside of the cell, so you just mix them together. It's one of the great things about electroporation. You just take cells, you take your plasmid, you mix them together, and you add a shock. And that makes the cell wall, the membranes kind of open up a little bit, so the DNA gets in, it recombines, and you can get a recombinant cell, or the plasmid goes in. And so it's a nice, simple way, because you just send a little electrical shock, and I think it takes about 20 minutes to do electroporation. The longest part is letting your cells thaw on ice. Protoplast fusion. So when you have plants and they get a wound, they have callus cells, which is what are basically stem cells in plants. And so that's how you can pluck a leaf off of some plants, put them in water, and they'll start to grow. Well, if you take that callus cell and you remove that cell wall, you're left with a protoplast. And so here you have this plant cell that doesn't have a cell wall. Well, if you take two protoplasts, you can merge them together, get them to become one cell, they'll build a cell wall again, and you'll be able to get an entire new species. So this is where a lot of those hybrids are coming from, is because species barriers are no longer an issue. We don't have to have broccoli and cauliflower mate. We just fuse a broccoli and a cauliflower together and get a cauliflower. That's how you get all those fun colors. Gene gun down the bottom. So, I mean, you literally coat little tiny 5 micrometer, nanometer gold particles, and you shoot it at the cells. And the idea is that maybe you might kill some in the first layer, but there's multiple layers of cells, and so Eventually, you'll get through the cell wall without going out the other end. And then micropipetting. This is how they'll do like, you can actually artificially insert genes, or if you wanted to take out a nucleus and put in a different nucleus, this would be how you would do it, would be the micropipetting. It's like how they would do Dolly and stuff. Do what? Dolly? How they created Dolly?
Clone sheep.
Way back when, there was a clone sheep named Dolly. You take,
I forgot her name.
you take the embryo, take out the nucleus, take the nucleus from a somatic cell. So what is a somatic cell? It's anything in our body that isn't a germ cell. Okay? Germ cells are ovaries and testes, the sperm and eggs. Those are germ cells. Everything else is somatic. So say they took, I think, breast tissue cell, I don't really remember, but took basically a somatic cell, fully adult nucleus, shoved it into the embryo, and that's how they got Dolly. So you would do that with a micropipette. Questions? No? Okay. So, other uses, DNA profiling. So, biotech has really brought DNA profiling along. It used to be you would need an entire knife tip, so a mill full of blood to do DNA profiling. Now you can actually use the backsplash on a gun, so a little tiny speck, because of PCR. So, but the basics with DNA profiling, you have either RFLP or PCR. So RFLP, restriction fragment length polymorphism. You basically are analyzing, you're cutting it, so restriction fragment length, you're cutting it with restriction enzymes, and you're looking at what are the fragment lengths. And polymorphism, many differences, where are those differences? So if you're able to take the defendant's blood, run it on a gel with, say, some blood you found on the defendant's clothes, and you run it against the victim's blood, you can start to see going, why is the victim's blood on the defendant's clothes? That's not his blood, it's their blood. And so this was kind of one of the first methods of actually being able to figure out how they're doing this. And so this is RFLP, it's restriction fragment lengths. You're looking at the different lengths of the DNA.
When they do this test, are they using anything besides the white blood cells, or is it just white blood cell DNA that they're testing?
Usually the white blood cells. I mean, you can do this with any cell. I mean, if they're doing it from blood, they're doing it from white blood cells. But I mean, you can do cheek swabs and stuff. So, but most of the stuff they do now is PCR because it's so much easier. So, RFLP and PCR. So, restriction fragment length polymorphism. So you analyze the pieces, you cut them, cut the DNA, run it on gel. PCR, polymerase chain reaction. So this is analysis of the DNA sample by making copies of your specific DNA. Okay, so we're going to talk about that one quite a bit. So, RFLP. We also use RFLP in microbiology to test microorganisms because their genomes are small enough that we can actually cut them up, run them on a gel, and see something. So this is something that, we don't necessarily use as much crime scene-wise, but we do use it as far as identifying organisms-wise. Because you can chop them up, digest their, so you isolate the DNA, chop it up with a restriction enzyme, and then you run it on a gel. You're able to get DNA fragments. And then at first, they all look the same. Or there's so many fragments, you're just going to see a smear. But what they do is they do a southern blot. So southern is DNA, and then so there's southern, western, and northern blots, but southern is for DNA. So you run your gel, and then you transfer all of these DNAs onto filter paper. So usually it's like nitrocellulose. And then you incubate. So you end up with this little piece of paper, this nylon filter, this nitrocellulose, which has all of this DNA transferred to it, stuck to it in thin membrane. And then you're going to wash it with probes that are usually radioactive. So you do a radioactive probe on there, and DNA is single or double-stranded?
Double.
Double-stranded. But by going through all of these steps, you've denatured it, so now it's single-stranded. So when you add probes that are also single-stranded, they'll bind and anneal, and so you'll be able to pick out specific sizes. And so instead of having this huge mess of DNA you're trying to look through, you can detect the probes, and then you can see very distinctly, oh, you know, A and B. You know, so it's like, if, you know, A was, this is, you know, people are getting sick, this is a sample from people getting sick. B and C could be two different organisms. You go, oh, organism B was getting people sick. You know, so it's a way to tell that. So that's a southern blot. So cut it up, run it on gel, transfer it to a nitrocellulose or nylon, you know, membrane, a filter, incubate it with single-stranded probes, detect. So you can either have them be radio labeled or fluorescent labeled. Either way to make them very visible. Okay. This is something they use for like sickle cell. Because if you want, you know, you can see the different cleavage patterns. You know, anytime you're messing with the DNA, that gives you a chance to have restriction enzyme sites. And so they figured out that normal cell DNA runs at one length, and sickle cell won't cut, so it will be a little bit longer. And so because of that single mutation, it changes how it will actually be cleaved, so you can run RFLP, cut them out, and then probe for it, because if you put your probe over here, it's going to get the ones that are, you know, the full length or the, you know, or half length. So you'll get both of them, so you can very clearly see, does somebody have sickle cell? Are they a carrier sickle cell? Are they homozygous for the normal copy? And so you're able to actually see what the genotype is based on this method. Okay. Does that make sense? Okay. Then because I think it's CODIS. Most of us have heard of CODIS. You've been living or exposed to TV lately. There's so many shows, crime shows. And so CODIS is just, they have 13 core STIR loci, so STR, short tandem repeats. So what they're looking at is they were looking for 13 places around the chromosome, around the genome, that have repeats. So they're little tiny repeats, and they just are repetitive. And what happens is, from person to person, the number of repeats is different. And so by looking at all the different repeats, you can actually tell who a person is. You know, and so they're in places that aren't necessarily as important, so that, you know, but they'll be maintained. So you can track. This is how they can do paternity testing. You can track how many of those repeats do you have. What is the likelihood? You know, it's like, so if you match up at all 13 loci, chances are that's the person who did it, or that's, you know, so and paternity testing can be very powerful just because there's only so many, you know, chances of not giving that exact. It's like genes, but they don't have any information. Okay, so they have 13 basic ones. What I thought was really kind of cool that they've discovered is that they can actually, oh, and then they also can do sex typing. Is they figured out these sizes, and then they used different fluorescent probes. So they came up with, they can put 4 to 5 different primers into a tube, and they can run all of them at once and come up with the reading. So they can get an entire genetic profile in one tube. And by making them different colors, you can see them on different spectrums. So, you know, even though, because these are blue, you'll set up, you know, primer here, you know, set up 4 different reactions. But you can do them all in the same tube, and since they're going to give you different sizes, it's very clear to tell, you know, this person is heterozygous at this location, they're homozygous at this location because they only have one spike. And so it's a way that you can actually go through and you can count, going, okay, they have 12 and 14 repeats. You or they have 28 and 30 repeats. And so for each position, it's just as good as, it's genetic information. So this is how they do the DNA fingerprinting. Okay. I always thought it was kind of interesting to actually see some of that.
That is interesting.
Okay, so PCR. What is this PCR we've been talking about? PCR, polymerase chain reaction, is the ability to go from one template DNA to exponentially amplify it into billions of copies. Okay, and so this is a great tool. It's kind of amazing. I think it's only about 30 to 40 years old. And so it has really changed the way we do science, just in almost every aspect, because you have the ability to amplify any piece of DNA. And then is this where and because it is so much easier to not write this on the board. Let's see how it's placed. Okay, so our first cycle, so PCR is in 3 stages. You have denaturation, annealing, and extension. So first, you heat it up to, I think they have like 95 degrees. You heat it up to almost boiling. That denatures the DNA, pulls it apart. So you end up with now single-strand DNA. And so the red and the orange are the sections that we want to replicate. Okay? So you heat it up, everything melts apart. And then, so that's at 95 degrees. Then what you're going to do is you're going to add primers. So these will lay down on your specific piece of DNA that you want.
It's the second step.
It's annealing.
Adding the primer is the second step.
Primers are the second step because you are annealing the primers. So you drop down to a nice cold temperature like 55 because you're trying to make it so these primers. So primers, just like when we talked about to make like the Okazaki fragments, you have those RNA primers. In this case, they're DNA primers because we've artificially created them in a lab. So you have these primers. They're just short pieces of DNA. Usually if you make them in a lab, you make them about 18 to 24 nucleotides, just because then it's only going to lay down in one spot. So, way hot, melt it all apart. Drop it down, you get your primers to sit on it. And then you heat it back up to like 72 degrees so that you can get extension, the third step. And so this allows you to get, to amplify up. So now all of a sudden we have 2 copies. But you might have noticed they're not exactly the right size. We only want the red and orange section. They're not exactly right. But as you go through multiple copies, you'll start to get just the piece you want. So, second cycle, heat it up, you anneal primers, you do extension, and you keep doing this. You do 30 cycles of this. And so you can still see that second cycle you only have 0 correct products, but you have 4 longer ones. It takes till the third cycle to when you can actually get the correct length. And what is great about this is that you artificially make these primers, which means you can put a restriction enzyme on the end of them. You can put a fluorescent probe. You can do whatever you want since you are creating them. But it allows you to amplify up just a piece of DNA. And by the time you get through 30 cycles, because we're only on 3, I don't know where the sound is. Aha.
At the end of the 5th cycle, there are 22 double-stranded DNA fragments of the correct length and 10 longer ones. Cycle 6, 10, 15, 20. After 30 cycles, there are over 1 billion fragments of the correct length, but only 60 longer ones. The product, therefore, consists of essentially pure target sequence.
Okay. I like it. I like that video just because it shows nicely how you can amplify up a ton of that specific sequence. This is why they can now do the backsplatter on a gun. That little drop of DNA is enough because you can amplify it. So we have the ability to just amplify up all the DNA we want, and then we can do all of our analysis. Okay? Any questions about PCR? What are the 3 steps?
What's the first one called?
Denaturation.
Oh, okay.
Denature, annealing, extension. Heat it way up, 95 degrees Celsius. What is boiling in Celsius?
100 degrees Celsius.
100. So we're really close to that boiling. Heat it up, cool it way down, anneal primers, warm it up, do extension. So the old...
Sorry, you said the first step was is
Denature.
basically denaturing. The third step was extension. What was the second step called in one word?
Annealing.
Annealing.
Annealing primers. So the way they used to do PCR is you'd have 3 different water baths. And so heat it up, you'd move it to let it anneal the primers, you'd add polymerase, because you need DNA polymerase to extend, just like you do, we're doing an artificial DNA replication, basically. So you add the polymerase, you put it in the water bath, and let it go. Then you heat it up again. Well, the thing is, if you're using human polymerase, we like 37 degrees. You heat up to 94, 95, and that enzyme's dead. Now what they've been able to do is they've gone to hot springs and pulled different polymerases out of Thermus aquaticus, which is what TAC is, one of the most common DNA polymerases. And so since it's used to living at a hot temperature, it likes 72 degrees Celsius, which is why we raise the temperature to 72. It's happy there. That's when it wants to work the best. But that also means that when we raise it to 94, 95 for briefly, it doesn't really care because it's used to being hot. And so it actually, you can just load it in, set it in a machine, and walk away. Make sense? Okay, so recombinant DNA technology. We talked about gel electrophoresis briefly, just running things on a gel. DNA is negative or positive? Charged. Negatively charged. So when you run it on a gel, you want to have it run towards the positive electrode. Okay, so that's gel electrophoresis. You're separating the DNA based on what? Size. Okay? It's gel electrophoresis. You load them in. It's easier. Okay. You have wells at the top. It's all underwater or under buffer. And so in here you're going to load your DNA, and then you have your negative pole, your positive pole, and so you're going to end up running a current through this, and the DNA is going to run from the negative towards the positive based on size. And so 5, 10, 15, 30, 40, 50 kb, kilobases, thousands of base pairs. Okay? So you always run a marker of some sort. Now, great thing with gel electrophoresis is you can then stain it. Usually, this is titanium bromide. Titanium bromide is an intercalating agent, so it gets into the DNA and then fluoresces when you put UV on it. And so you can actually see, okay, here's a nice ladder, here's your DNA bands, here's just random smear, it happens, but you can actually separate things based on size. And so you can either separate it by size and take a picture of it if you want it for analysis, or say you were trying to, like for the bovine, to get growth hormone, you could cut up your genome and you want that certain piece, you could actually come in and cut it out. And then you just melt it out of the gel, and now you have purified DNA that's exactly the size you want. Okay? What else do they want?